In this video, we're going to talk about monoclonal antibodies. These are important because they are a targeted way, especially to combat cancer. And, chemotherapy and radiation have had some success, but they have so many side effects that if we can get a more targeted treatment, this could really help the quality of life of the patients. And so it's kind of complicated how they make these antibodies. I'll try to simplify this. But first, they will use a mouse typically, and they'll inject an antigen into the mouse, a specific antigen that they want to have a specific antibody for. And they'll give the mouse a couple of weeks to build up the antibodies. And when they have a maximum amount of antibodies, they found that once they get these antibodies and they put them in a culture, they will not replicate normally because basically they've taken the plasma cells, the B cells that make antibodies, out of the spleen. So they'll euthanize the mouse. They'll take the spleen antibodies out, or the cells that make the antibodies out, but they will not crank out the antibodies in a culture. So what they found is if they use cancer cells, cancer cells are known to cause proliferation, and myeloma cells come from multiple myeloma patients, and they are plasma cells. So they're B lymphocytes that have differentiated into plasma cells, and those are the cells that make antibodies. So we have two types of lymphocytes. If you remember, we have T lymphocytes and B lymphocytes. The only ones that can make antibodies are B lymphocytes, and they mature a little further and differentiate a little further into plasma cells when they do that. So they'll use these cancer cells, and they found that when they can make a hybrid of the two, bring in the antibody that they want to make a lot of, essentially, with a cell that will cause production of that antibody, ultimately you can get a large amount of them and produce them and use them for medical purposes. And that's kind of the gist of the rest of that. So how they work, just like any antibody, for example, if you have a cancer cell, it has antigens on the surface of the cell membrane, and if you have this specific monoclonal antibody, it can go and find that antigen on the cancer cell. These antigens wouldn't be on our normal cells, so it's a targeted therapy, and it would tag that cancer cell to be killed by another cell. So this is an indirect method that these monoclonal antibodies kill cancer cells. So here's kind of a review. We have the antibody made by the mouse. They take it out of the mouse's spleen. They bring it in with this cancer cell so they can produce a bunch of them. Then they use them, and they target the type of cancer and the antigens that those cancer cells have. They'll bind to those cancer cells, and it's like setting off an alarm for the effector cells to come clean house and kill these cancer cells. So you have natural killer cells, NK cells, T cells, specifically cytotoxic T cells, because we have two different kinds, and macrophages, which mean big eaters. So these three cells, once they see those monoclonal antibodies have tagged the cancer cell, they will come and kill the cancer cell specifically, and our cells are spared. So I want to give you one example of a monoclonal antibody that's had a lot of success medically, and this is called Herceptin. That's the trade name. Trastuzumab is the generic name, and you can see it there, the generic name written out for you, Trastuzumab. And all these monoclonal antibodies, you'll always see they end in umab. So that's kind of a hint that if you see a generic name of a medication and it ends in umab, you automatically know you're talking about a monoclonal antibody, and these will be more and more prevalent. Their research is heavy in this area because we're finally starting to be able to target treatments with cancer. So with breast cancer, 25% of breast cancers have a mutation in a specific gene. And genes that can become cancerous are called oncogenes. So in this oncogene, if they have a mutation in this particular one, it causes overexpression of a certain receptor in the cell membrane of the breast cells. And it's called the human epidermal growth factor receptor. So it's HER2 for short. And what that is you have growth factors that will bind to these receptors, and it causes proliferation of the cells, mitosis of the cells. And if you have overexpression of this particular receptor, you're going to have overexpression and proliferation of those cells, overexpression of the proliferation of the cells, and that's going to be out of control mitosis. So in this sense, it's not going to kill the cancer cell, but what it does is, let's see, there we go. So what it does is it will actually bind to the receptor and competitively inhibit anything else from binding to it. So growth factors, the human epidermal growth factor, will come and try to bind to it, but if you have those antibodies blocking the receptor, then they can't bind to it and they can't have their effect. So for example, at the top, that might be a normal cell with just one receptor, and then the bottom would be overexpression of these HER2 receptors. And that means that they're going to have a more pronounced effect because they're going to have more binding to growth factors. So Herceptin is basically intercepting the growth factors' influence on those receptors. So trastuzumab is the generic name. Herceptin is the trade name. It's used to combat certain types of breast cancer. And they'll do a genetic test to see if the patients can benefit from this. And it can either treat the cancer exclusively without having to have any chemo or any radiation, or it can be used as an adjunct to chemo and radiation and decrease the dosage and have a much better prognosis and less side effects. So that's a couple different ways that monoclonal antibodies can help with cancer. One is directly killing the cancer cell, not directly killing the cancer cells, but binding to the cancer cells, and indirectly killing the cancer cells by alerting the effector cells, the natural killer cells, macrophages, and cytotoxic T cells to come and kill the actual cancer cell. Or they can block receptors that when they bind to their ligand, which in this case is the growth factors, they cause cancer. Then if you block that from occurring, you block the source of the cancer from occurring. So it's really exciting in the medical field, and it's something that shows a lot of promise, and I'm excited for cancer patients that some of them have this option. And we'll talk to you in the next video.